Okay, if you want, we can start.
Oh, okay. Sorry. Yeah.
Yeah. Hi, everybody. Welcome to our seminar or webinar. My name is Nancy Hakouz. I am a professor at the University of Jordan School of Pharmacy. And we are welcoming today Professor Jesse Swen. Jesse Swen is a PharmD, and he is a full professor of the clinical pharmacy, in particular translational pharmacogenetics. He works as a clinical pharmacist, clinical pharmacologist at the Department of Clinical Pharmacy and Toxicology in Leiden University Medical Center, where he is the chair of the laboratory of the hospital pharmacy. His goals, his career goals, is to improve the outcome of drug treatment by gaining a better understanding of the genetic mechanisms that result in inter-individual variability in drug response. To this end, he's working on the many research topics. For example, dissecting the impact of rare, structural, and regulatory variants on drug absorption, distribution, metabolism, and excretion. Elucidating the mismatch between the drug metabolizer genotype and the capacity of an individual to metabolize a drug, which basically we're talking about penoconversion, translating pharmacogenomics to patient care. Now, he has authored more than 150 articles in international peer-reviewed scientific journals and several chapters in books. He is one of the primary investigators of the ubiquitous pharmacogenomic project and the leader of the PREPARE trial that assisted the clinical utility of pharmacogenetic panel testing across 7 European sites. In addition, he is an active member of the Dutch pharmacogenetic working group and the US Clinical Pharmacogenetic Implementation Consortium, CPIC. The title of his talk today is the PREPARE Study, Results and Future Perspective. The floor is yours.
Thank you, Nancy, for this kind introduction, and thank you for the invitation to present here to all of you remotely. It's an honor to do this. And I'm happy to tell you about our work in the PREPARE study and translating pharmacogenetics to clinical practice. I hope you can all see my screen. Is that
Yes.
still working?
Yeah,
all
it's
right.
working. Yeah.
So then I think to start, I thought, okay, I have some time. So this is actually where I work. I would like to show it to you. This is the Leiden University Medical Center. And this is the oldest main central building of the Leiden University, which was founded in 1575. So on the one hand, we still have these very old traditions. This is the annual opening of the academic year, where all the professors walk through the city from this building to a central facility. But we also have a very modern facility. So this is my lab, which is an ISO 15189 and GMP accredited lab in the pharmacy. So that is my background. So to start at the beginning, why pharmacogenomics? So I'm a clinical pharmacist, pharmacologist by training. I work with drugs all the time, and it's good to realize that drugs are, in fact, the most frequently applied medical intervention. And of course, they work, otherwise we wouldn't have them. But sometimes response is not as good as we would want it to be. And for example, if you look at classical chemotherapy, response rate is only 25%. And of course, we would like this to be a little bit better. Same, for example, is the truth for the treatment of depression with SSRIs. It can take weeks to months or even longer to find the right treatment for a particular patient. So on top of not working for all the patients, these drugs, almost all of them, do cause adverse drug reactions. And this is also an important problem because data again and again show that 6 to 7% of all the hospital admissions, in fact, are drug related, and half of those are the direct result of an adverse drug reaction. And the question now, of course, is how do we solve these problems of non-response and adverse drug reactions in clinical practice? Well, that is what this cartoon very nicely summarizes. We prescribe according to standards or to clinical guidelines, and if it then doesn't work, we change the dose. Well, of course, from a patient perspective, you would like to get the right drug straight away. For this variability in drug response, there are many reasons, and this is what clinical pharmacology studies and also clinical pharmacy, of course. And I think that we should take into account all these reasons, but I personally believe that by using our knowledge of genetic variation and how this influences drug response, we can do a much better job, and we can really improve the outcomes of drug treatment. This idea, the concept of pharmacogenetics is very straightforward. Before you prescribe or dispense a drug, you run a DNA test, and based on the test result, you either adjust the dose of the drug or maybe the drug selection. And this idea is not new. It was coined already at the beginning of the 20th century. At the time, it was called chemical individuality. And it has been around as a promise for a very long time. And sometimes I have the idea that this even holds back pharmacogenomics, because it has been a promise for so many years that everybody believes in it as a concept. But everybody also thinks, okay, maybe in the next 20 years or something. And that is not where we are right now, according to my feeling. I think that in the last 10 to 15 years, and also with the UPGX Prepare study, we have taken significant steps forward, and we were really at the moment where we can start implementing this and use actually what we have to improve the outcomes of drug treatment. And that is what I also want to show you in my presentation. Where are we? What have we learned from Prepare, and how can we move beyond that? But before I get to Prepare, I would like to show you one drug, one gene example to illustrate of what you already can do by using pharmacogenomics. And this is about 5-FU and capecitabine. These are widely used anti-cancer drugs. They work, but well, again, they have also caused a lot of toxicity. Most importantly, up to 1% of the patients dies of the treatment. So not of the cancer, but really of the treatment. This is a drug-gene pair where pharmacogenomics can really help to prevent this. So if you look at the pharmacokinetics of 5-FU and its oral prodrug capecitabine, then you can see that most of the 5-FU is actually, most of the 5-FU is converted into inactive metabolites by an enzyme called DPD. A small proportion is converted into active metabolites, and that effect has that part has the anti-cancer effect. When you look at this drug-gene pair, it was known that that approximately 5 to 6% of all the people have a non-active DPD enzyme, and that this is partially caused by genetic variants in the gene encoding DPD, which is called DPYD. And you can imagine that if you have such a variant, you have no active DPD, and you have a much higher exposure to these active metabolites, and a much higher risk for toxicity. And the patients who have these variants, those are the ones who are at risk for fatal toxicity. So this was known actually for 10 to 15 years, I think, maybe even 20. And then a group in the Netherlands from the Dutch Cancer Institute did a first study investigating the DPYD STAR2A variant that showed that if you did a pharmacogenetic test and treated the patients with this DPYD variant with a 50% reduced dose, then you could actually prevent a large proportion of this toxicity. And based on that study, we designed a follow-up study, which was called the ALT-DPYD project. It was a collaboration between Leiden University Medical Center, Erasmus Medical Center, and the Dutch Cancer Institute. And we ran a large prospective trial in 17 hospitals in the Netherlands that enrolled 1,181 patients that were all tested for 4 genetic variants in DPYD. And if you carried these top 2 variants, then you received a 50% dose reduction. And if you carried one of these, then you received a 25% dose reduction. And all reduced dose were in subsequent treatment cycles were ramped up according to the occurrence of toxicity. And what we showed in this study is actually that by doing so, you could, let's say, reduce the fluoropyrimidine-related toxicity, the relative risk, from 2.87 to 1.31. So this is a very good result. It showed that you can actually use this pharmacogenetic test to reduce the amount of adverse drug reactions. You could prevent all toxic deaths. And when you did the calculation, we could also show that it was in fact a cost-saving strategy. So based on these results, researchers from the Dutch Cancer Institute made an appeal to the EMA, and they asked them to include this information in the drug label. And after internal discussions, the EMA in April 2020 released this piece of information, this warning, that they recommended DPD testing, so not a genetic test, but the DPD activity test. And they explained that you could either do that by a genetic test for the 4 variants or an enzymatic test. And this has resulted in an enormous uptake of this test in Europe. So these are the results from a survey that actually asked people about their use of DPYD testing prior to the EMA warning and the year after the EMA warning. And what you can see here is that overall in Europe, there was a 76% increase of pharmacogenetic testing for DPYD. And I'm quite convinced that if you would repeat this survey now, that the increase would be even stronger. A big discussion surrounding this test has always been the question that we received a lot from oncologists that asked us, okay, so you have a patient with a tumor and you run a DNA test, and based on the test you find genetic variants, and if someone carries the variant, then you reduce the dose. Of course, you see less adverse drug reactions because you reduce the dose. If you would not give any drug at all, there would be no toxicity. And that was one point. And the next point then was, we are concerned about the efficacy because you reduce the dose. Is it still effective? And we very recently published the results of that analysis. So we collected a survival of all the patients who participated in the study, and we specifically looked at the variant carriers. So we had 156 DPYD variant carriers and matched them with controls. And we run a matched pair analysis. And here you see the results for the 4 different variants and the 4 different panels. You can see that here's the STAR2A variant, the 2846 variant, and the 1236 variant, and this is the combined result. here for PFS and here for OS, and you can see that in fact everywhere the lines cross, so there is no difference in survival, with the exception maybe for the 1236 variant, because here there appears to be some trend in lower progression-free survival for people who received a reduced dose. But overall, survival is the same, and also when you do the pooled analysis, there is no difference. So this, to our findings, shows that you can apply DPYD guided dosing safely without compromising effectiveness. This is one example of a one drug, one gene pair that can help to improve the outcomes of drug treatment. There are many other examples, and we don't have time to go through these in detail, but I just want to show them to you. And these are all very well evidenced gene-drug pairs where it has been shown that the pharmacogenetic test actually is useful and is a clinical useful test. So based on this, the question arises, okay, so we have this evidence for single drug gene pairs, and on top of that, we have guidelines that tell you exactly how to change the dose, how to respond to the results of a genetic test in guidelines from the Dutch Pharmacogenetics Working Group, and also from the CPIC and from Canadian and French groups. We also have some knowledge on the potential impact of pharmacogenetic testing, because we know that if you test large groups of patients for the genes covered in these guidelines, then you will find that 95%, almost everyone, has at least one variant for which already a recommendation is in place. And if you look at the drugs in these guidelines, you will see that these are not rarely used drugs. No, they are very commonly used. So we have common genetic variants, and we have recommendations for drugs that are in fact frequently used. So the question then arises, what's the impact of a pharmacogenomic panel test? Why test one gene? Why not test the entire panel? And the only thing here is, so this is really the next piece of the puzzle, but convincing clinical data supporting such a panel test were lacking. And that's why we designed the Ubiquitous Pharmacogenomics project. It received 50 million euros funding from the EU to investigate prospective real-world implementation of a pharmacogenomic panel test combined with the Dutch Pharmacogenetics Working Group guidelines in 7 EU countries. Here you can see all the countries that participated in the project. The countries that actually implemented pharmacogenomic testing are Italy, this is Greece, of course, Greece, Italy, Slovenia, Austria, the Netherlands, Spain, and the UK. So we have implementation in 7 different countries, 7 different healthcare settings. So there was enormous variability between these countries in the healthcare system, but also in the level of implementation of pharmacogenetics. And therefore, we developed a set of tools to get everyone on the same level. And the first tool were, in fact, the Dutch Pharmacogenetics Working Group guidelines. Here you can see some of the guidelines that we have developed. So in orange, the genes, and in black, the drugs, which we reviewed. And you can also see how this is implemented in the Netherlands. Because in the Netherlands, if you record, for example, that you are a CYP2D6 poor metabolizer at your GP or at your pharmacy or in your hospital, then as soon as there is one of these recommendations relevant for the drug you are prescribed, in this case clomipramine, then on your screen the doctor or the physician gets a pop-up message. It's important to say that these are evidence-based guidelines. So the group was founded in 2005. We have done systematic literature review, and for each drug gene pair and for each particular phenotype, we have calculated a dose adjustment with the objective to obtain similar exposure as the normal metabolizer. And this is a nice illustration created by Julia Stingel many years ago, but it shows a number of drugs, the percentage of the dosage needed to get the same exposure as a normal metabolizer, and then you see the dose for the poor metabolizer, the intermediate, the normal, and the ultra-rapid metabolizer. And you can see here that for Venla vaccine, for example, a poor metabolizer requires a 9-fold lower dose compared to an ultra-rapid metabolizer, which is an enormous difference. These Dutch Pharmacogenetics Working Group guidelines for the UPGX project, they were translated into English and then in Greek, Spanish, German, and so forth and so on. And these guidelines are also available on PharmGKB website, and there you can see that they are they have this nice orange color, which is on which you can recognize it. And, oh yeah, and I forgot to say, of course, there is also the CPIC guidelines, but we investigated a couple of years ago the differences between these two sets of guidelines, and there are differences, but in general they are quite comparable. The second tool that we had was actually our pharmacogenetic passport. We started with genes covered in the Dutch Pharmacogenetic Working Group guidelines, and we found out that there were 12 genes at the time with an actionable recommendation. And then we looked at the minor allele frequency to select individual variants. And in the end, we had 50 variants in 12 genes that were included in our pharmacogenomic passport. The third tool, that was a delivery tool. In the Netherlands, we have a very nicely electronic system that I just explained to you, but this was not the case in all the countries, and therefore we developed the medication safety code card. This was originally designed by Professor Matthias Samwald from Vienna. It's this credit card like thing with a QR code. This is my card. If you scan it, then you see my pharmacogenetic test result. So I have a variant in DPYD. So if I ever were to develop colorectal cancer, for example, and to be treated with capecitabine, and this is in Dutch, apologize, then I should receive a 50% dose reduction. But this is a very nice system that makes it very easy to have the pharmacogenetic recommendations and the test results available at point of care. So with all these tools, we were able to conduct the clinical study, which was called PREPARE. This was an open label, multi-center controlled, cluster randomized crossover implementation study of this 12 gene PGX panel combined with the Dutch Pharmacogenetics Working Group guidelines. We had centers in 7 different countries. Patients could be enrolled if they started with a first prescription, so really a new prescription for one of the 39 drugs with actionable recommendations in the Dutch guidelines. And because we had 39 different drugs, we were limited in the endpoints that we could study. So efficacy was not an option because with drugs ranging from anti-tumor to antidepressants, there is no universal efficacy outcome, but all these drugs do cause adverse drug reactions. So we focused on the occurrence of causal clinically relevant adverse drug reactions caused by the index drug in the first 12 weeks of the study. Here you see the patient journey in the study arm. When a patient started with the drug and provided informed consent, a DNA sample was taken. Within 7 days, the genetic results were returned and recorded in the EMR. And from that moment on, you had pharmacogenomics guided prescribing. Patients received a medication safety code card, of course, and we collected at set time points of 2, 4, 8, and 12 weeks information about the occurrence of adverse drug reactions. And this was either done by a research nurse or by a digital survey. So all these data were collected and then filtered. These ADEs had to be of a certain level of severity, and the causality had to be at least possible. And this was assessed systematically by using the Liverpool Causality Assessment Tool. If this was the case, then the ADR contributed to our overall clinical endpoint. Because these scores were all done by local study teams, and this was an open label study, we wanted to protect ourselves against bias, and we took a random 10% of these assessments, and we sent them to the Dutch Pharmacovigilance Center LAREP, and they did a reassessment blinded for the arm allocation, and these results very well match with the original scores. So then we have some results here. You can see the patient flow, and in total we had 6,944 patients, of roughly half in the control and half in the study arm. On the top here you can see the patients that were assessed for eligibility and the patients that were enrolled. And when we go further down to the bottom, you can see the patients that had an actionable genetic variant, so an actionable drug-gene interaction for their drug of inclusion. And this is in the control arm and in the study arm, approximately 25% of all the patients in that arm. You may also see that in the Netherlands, there is something different compared to the other countries, because the number of patients that were assessed for eligibility is much higher, and the number and the percentage of enrolled patients is much lower. And this has to do with the fact that in the Netherlands we use 28 community pharmacies and a digital algorithm from a Dutch company called nControl to flag patients that were eligible for this project. So this is the explanation of these different numbers. In our statistical analysis, we decided to perform a gatekeeping analysis. And in this gatekeeping analysis, it allows you to do multiple tests, pre-specified tests, without losing a lot of power. And we decided to first compare the patients with an actionable drug-gene interaction in the study arm with the actionable patients in the control arm. And this was done because in these patients, you expect the biggest effect, because here you're actually making a change. And only if this was significant, we were to compare the full study arm and the full control arm. Here you can see, irrespective of the drugs that were used, you can see the genes and the actionability. So overall, 93.5% carried at least one actionable variant. Most actionable genotypes were found for CYP26, which was to be expected. And you can go down here in the list and you can see, for example, that for DPYD, this was much lower. But overall, this is very much in line with what we see also in much bigger trials where they look at pharmacogenetic variants. If you then look at the drugs on which patients, the index drugs on which patients were enrolled, and the actionability, then you can see that, as you may expect, most patients were enrolled on atorvastatin, and also simvastatin was very frequently used. Tacrolimus was frequently used because in Vienna we had a transplant clinic who almost exclusively included kidney transplant patients, all treated with tacrolimus. In blue, you see the number of patients. In orange, you see a percentage that was actionable. And then here you see the unfiltered adverse drug events and the severity scores. And you can see that there was considerable heterogeneity between the countries. And also this was expected because, for example, in Italy, the most adverse drug events are reported but that was also a tertiary cancer clinic, so they apply way more toxic drugs than, for example, what was done in the Netherlands. To reiterate, we only counted adverse events of grade 2 and higher, so orange and higher, with the exception of Italy, because there we used so many toxic drugs that we decided to only use grade 3, or for hematological toxicity, even only grade 4 or higher. So the PREPARE study, the primary endpoint, this is the results in actionables. Here on the y-axis you see the event rate, and here you see the control in the study arm. And what you can see is that the incidence of these clinically relevant adverse drug reactions dropped from 0.28 to 0.21, and that this difference is statistically significant. And this is, of course, a fantastic result. We were very happy when we first saw the results of this analysis, because this is what we had hoped to find. We then moved on to the second step of our gatekeeping analysis, comparing all the patients. And there we found a highly similar result. Here you see the drop from 0.29 to 0.21 in the study arm. The difference is the significance level is even better. That has to do with the larger numbers. But this is not what we had expected, because here we had expected a diluted effect, because in this comparison also the patients are included that have had no actionable genetic variant. And we explored this in more detail. And what we have come to learn is that actually it's changes in case mix that drive this effect in all patients. So what do I mean with that? Well, here you can see again a little bit the design of the study. So this is one country, and we have different centers recruiting patients. We have PGX guided prescribing, and then after crossover, we have standard of care, and you see patients with a low toxicity profile and with a high toxicity profile. And by the way, this is just an illustration. This is in the ideal world. However, it was a real world study, and changes were made. So there were additional centers, recruitment centers, and there were changes in drugs and prescription patterns. And also we applied capping. So what happened was that actually in some of the countries, these arms were not comparable, and there was a disbalance. We needed to correct for that. And we did so in a post hoc exploratory analysis. And if we do so, then you see that in fact the effect in actionables increases a little bit. So the odds ratio becomes a little bit bigger. And in all the patients, when you compare all the patients in the study and the control arm, the effect loses significance and the odds ratio decreases. And what you can also see is that, in fact, it's really the changes in these drug prescription patterns that correspond to the effect in the non-actionables. So our conclusion is really that there is an effect of pharmacogenetic panel testing, and that this effect in all the patients is due to these changes in case mix. Well, this was published in Lancet. It generated enormous impact. It was discussed in, it was the main news in many European countries, in the Netherlands also. And it was discussed in the scientific literature. There were critical letters, mostly referring to the lack of blinding. This was an open label study, and also to the lack of economic analysis. But there were also very good, or very positive, I should say, reviews, for example, here in the Annals of Internal Medicine, where it gets a very high clinical impact rating. And the bottom line conclusion is that pharmacogenetic panel testing actually works. And then it also generated a lot of discussions about how to implement this. So here you can see the Dutch healthcare minister at the time of the study last year, and there were questions in Parliament: should we now run a pharmacogenetic panel test for all Dutch citizens. And here he is explaining that, yes, it's a very good study, but no, I unfortunately cannot afford to run this test in all Dutch citizens. So he actually made the point that we should have more data on the cost effectiveness and also data on for which group a pharmacogenetic panel test is most beneficial. Another effect of this publication in Lancet was that there was a lot of public awareness of pharmacogenetic testing, and this is from a German game show where Weisdenser was. It's a form of Weekend Millionaires, where in May last year there was actually a question about pharmacogenetic panel testing. So the question is, how can you reduce adverse drug reactions? Is it by coating tablets with a fat film? Is it by taking a 2-hour break, or is it by doing a pharmacogenetic test? So this is, I think they got the answer right. So they earned 1,500 euros, which is a bargain for a 50 million euro project. But it nicely illustrates the enormous societal impact of the project. We have been working on cost effectiveness analysis since, and 3 country-specific analyses have been published. They all show favorable results, and we have almost finished the overall economic analysis. So you can really expect that to be released soon. So where are we then with pharmacogenetic testing? Well, I think we have gone a long way. We've gone from bench to bed and maybe also back again. So many years ago, we identified barriers for implementation. We have worked on providing evidence because that was an important barrier. We now also have evidence for panel testing, and we're really at this point where we should move this into the clinic. In the meantime, we also know that our tests are not perfect, and there is definitely room for improvement. So there are scientific challenges there. And also there are health policy challenges. So with regards to the scientific challenges, I think it's important to realize that we have tested a 50 variant panel in 12 genes, which is next to nothing if you look at the total variability in the genome. And this is a nice example. I like this example where they have shown that to explain the genetic variability in height, you need to look at more than 12,000 individual single nucleotide variants, and to identify those, they needed a sample size of 5.4 million. Well, compare that to our 50 variant panel. So there is really a lot of room for improvement here. We need bigger studies, and we need to do a better job there. But the good news is that there is an enormous technology drive because our ability to interrogate the genome are continuously improving. Another thing is that we need to do a better job at genetic diversity. So most of the data that we have originate from people of European descent, and well, there is a lot more genetic variability in the world, and this is a sad example of the impact that may have. So what you see here is the Kapoor family. Most of them work in medicine, maybe all, and they live in Canada. And this is their brother, Anil Kapoor, who actually was a surgeon and had colorectal cancer, or colorectal cancer, and colon cancer, I think. And he, in a few weeks after starting treatment with 5-FU, he passed away. And the thing is, he had received pharmacogenetic testing for the 4 variants that I earlier discussed. But after he died, they did additional sequencing, and they found that he carried a variant that is quite common in India, but not so in Europeans. And as his family originates from India, well, this might have potentially been prevented. Another thing is that when you look at it from a research perspective, we are trying to relate genetic variation to drug response here at the bottom, right? And we are ignoring all the intermediate steps. We don't have the time to discuss these in detail, but in green, there are all the opportunities to do more discovery work here, to do better discovery work. So I think there is a real challenge there. And another challenge is that we need to integrate our pharmacogenetic test within all the other clinical pharmacology factors that affect drug response. So we could do that better. With regards to prepare, we are doing additional analysis. So we collected all these data on adverse drug reactions. And for example, colleagues of mine have been working on one specific adverse drug reaction that was reported, and that is dysgeusia, so the loss of taste. And in the Italian cancer patients, this was quite frequently reported. And there we did an exploratory GWAS focusing on dysgeusia. And unfortunately, we didn't find genome-wide significant hits, but we did identify a number of potential variants that are suggestive for statistical significance, so they're above this line, and we may want to investigate those further. Another thing is that we have been working on not on array-based discovery studies, but using advanced sequencing techniques such as long-read sequencing combined with machine learning approaches to elucidate complex pharmacogenetic regions. And in doing so, for example, we have showed that by using long-read sequencing combined with machine learning, we, let's say, can improve the explained variability of CYP2D6 activity from 42% to 79%. So also here, I think this is an enormous scientific opportunity to do a better job of pharmacogenetic testing. And finally, we have after the UPGX project has been completed, we have a follow-up project or a spin-off. Now it's a really follow-up project, which is chaired by Thorsten Lehr from Saarbrücken in Germany here. And in this project, we want to integrate drug-gene interactions with drug-drug gene interactions. So really focus on the composite drug-drug gene interactions. And if you want to read more about this project, I would recommend you to go to this website or contact me or Thorsten afterwards. When you look at clinical practice, there is another development ongoing, and that is really preemptive pharmacogenetic testing. And this particularly relates to the fact that in the clinical genetics world, people have moved away from testing single genes into performing whole exome or even whole genome sequencing. And we've worked together with these clinical genetics groups to actually investigate if you can repurpose these whole exome sequencing data for a pharmacogenetics profile. And we published about this, and we showed that with the exception of some variants in, for example, 2C19 and also in 2D6 copy number variation, you can actually really do this. And I think that we will see more and more this repurposing of pharmacogenetic test results, or no, I should say repurposing of genetic test results for pharmacogenetics in the coming years. So then there is some health policy challenges. We already talked about cost effectiveness. This is of course very important, and the need to identify which patients benefit most of pharmacogenetic testing. If we have those data, then we can ensure reimbursement for the pharmacogenetic test. Then we have the issue of knowledge. So training and education is very important. We put a lot of effort in that in the UPGX project, but I think also in the clinical implementation of pharmacogenetic testing, this is something that can be improved. And the same holds in fact true for the infrastructure. because the tests will be there. The infrastructure to do the tests is available in most countries, but the big challenge is actually the exchange of the results in the entire healthcare chain. It makes no sense to retest all the patients again and again, because this is a DNA test, and we should really have systems in place that allow us to share the results. And then finally, with regards to health policy, it's also important to mention the regulatory impact. And I've shown you this effect of the EMA label change. And it's very interesting to know that in the last couple of years, we now have seen that there are, in fact, 4 drugs that have mandatory pharmacogenetic tests available in their drug label in Europe. And they were on this slide. So I think that's also a big step forward. And finally, and this is in Europe, this is another, I think, important project. We have the 1+Million Genomes initiative. This is already ongoing for quite some years. It will at least run until 2027. And in this project, a lot of patients will be sequenced. So recently, 45 million budget was allocated to sequence a large group of patients. But what is most interesting is that below the umbrella of this project, there are use case working groups, and this year there was a new one, and that is a use case group on pharmacogenetics. And I think this is also a very important development for pharmacogenomics in Europe. And with that, I would like to finish. So what I've shown you, in fact, is that this implementation of pre-therapeutic pharmacogenomic panel testing is feasible and effective. It reduces the number of adverse drug reactions. However, we still have a lot of challenges. We can do a better job. We have scientific challenges and health policy challenges. But I'm happy to say that there's also a lot of opportunities. And overall, we can see that pharmacogenomics is moving from reactive testing, so if the damage has already been done, to pre-therapeutic and also preemptive panel testing. And I think that is a very good development. So ultimately, I hope we can make this cartoon where let's say the prescription has been individualized based on the pharmacogenomic profile and the Dutch or other pharmacogenetics guidelines. And with that, I would like to end my presentation. I would like to thank all the patients who contributed to this project, and of course all my collaborators and you for your attention. And I would be happy to take any questions. Thank you.
Thank you, Jesse. A very insightful presentation. I think having a reward evidence that pharmacogenetics works, it might pave the way for other countries to try and to get this into the actual clinical practice. Now, I don't have a lot of questions in the question and A session, but I have questions in the chat. There's a question from Andre. I can't pronounce the second name, Senegi. How ready are we to have the barrier-free clinical decision support system for all, at least all cancer patients? And how long does it take to have the information ready?
So I think this medication safety code card system is the inventor of that. So Matthias Samwald did a very nice job in making that an open source system. So that is, in fact, it is available. You can just use it. The challenge, of course, is to have the pharmacogenetic test available, and that is the big challenge. There is well, I don't think it's not the test itself, because it's a relatively straightforward genetic test, so you don't need very expensive machines to do it, but you do need the knowledge, and of course you need the reimbursement, right? So that is, yeah, and that is at least in my experience in the Netherlands, that is a challenge here to get everything reimbursed. With regards to if you're referring to how long does it take to get the test results back, yeah, in the practice, the way it's organized here in the Netherlands, that's really fast. So it's usually within, if you do a single gene test, it's definitely if it's in our institute, it's usually 1 or 2 days. If it's outside our institute, because we then have to ship samples, it always takes a little bit more time. And if it's a panel test, then also it takes a bit more time. So that is still 2 weeks. Yeah.
Can I ask about the card information?
Yeah.
Is it updated if the guidelines have been changed?
Yeah, so that is that's the nice thing of this QR code system. So we in the back end of the card, we can, let's say, update the recommendations, and that's also what we do. So the genetic test results stay, of course, the same, but sometimes the recommendations change, or there are additional variants, and then it depends on which genetic test was done to let's say create the card, because I didn't have the time to tell that, but I mean, like, we could produce the card based on whole exome sequencing or whole genome sequencing, so ideally whole genome sequencing, and then you could also update the genetic test results. So that's not in the open source version of the card, but that is something that we have developed. But we also use the card with a small panel test, and then of course you cannot update the genetic results because you only have these 50 variants, for example, but you can update the recommendations. Yeah.
Yeah, okay. Somebody, Dr. Zaina Mahari, was asking about DPYD testing and the protein testing. Do you think there is a difference between testing the actual gene or testing the activity of the protein?
Yeah, this is a very good question, and this is actually an ongoing debate. So of course, I would say it at first sight it appears more logical to test the real enzymatic activity, right? Because that is what you're trying to find. However, it's my experience that in the enzymatic test is a lot more challenging, and there are several ones. So you can measure uracil levels. This is the natural substrate of DPD. You can also do an enzymatic test. The enzymatic test is very expensive and challenging, but it's the best one. It's absolutely the best one. But I don't think it's very feasible to implement on a large scale. The uracil test also is more closely to measuring enzymatic activity, but we have just completed a very large national study with that test, where the results were not reliable enough. So the problem is that the pre-analytical validity of the test is very challenging. So if you have you take a sample, for example, and you put it on the bench and you leave it there for 6 hours, then the uracil level is completely unreliable. So that also there is challenges with the execution of the test. But in France, this is the standard. That brings us to the genetic test, and the genetic test has the advantage that it's very stable, it's easily performed, but it has the limitation in a way that it's limited at the variance of which you look. Yeah, so there is a limitation there.
I think a lot of countries, they use the actual enzymatic activity in their practice rather than the genetic testing. And yeah, there's a comment in the chat from Andre saying that he's been in pharmacovigilance for almost 2 decades, and he sees personally the benefit from the decrease of the ADRs by help of gene testing. So he's agreeing with your findings.
Yeah.
There's another question from Mustafa. He said, thank you for the very informative presentation. So from the government perspective, I don't know if you can answer the government, would the pharmacogenetic testing be cost effective if the genes that contribute to health problems are not very common?
Yeah, that is a very good question. Of course, so if you focus on the genetic panel that was used in PREPARE, there the variants are quite common, right? And I'm absolutely convinced that we will be able to show that it's cost effective to perform this test for large groups of people, maybe even everyone. If you look at the rare variants, which of course are there, then I think it's a bit more tricky. It really also then depends on the adverse drug reactions that you are trying to prevent. But when you take a lifetime horizon in your cost effectiveness analysis, I'm sure that almost every test will show that that's beneficial, because I mean, like the costs of a pharmacogenetic test are, what is it, 300 euros for a very big panel test, and then you with almost everyone will in his over lifetime will start using drugs for which we have recommendations. So I don't think the problem with cost effectiveness is that we need data from 1 or 2 well-performed studies, and then there will be no problem with cost effectiveness anymore, because I'm convinced it will be cost effective, but we need to show convincingly. Yeah.
I think coming back to what you said at the beginning, that we know, and the evidence is there, that pharmacogenetics is very important. And we are still talking about this, part of it, because it's technology driven. And it was very expensive to do any kind of pharmacogenetic testing before. Now you can do it much cheaper. And maybe this is the reason why it's not being implemented, or it has been part of the normal clinical decision support systems. There's another question about DPWG. Is it in Dutch? All the website. It's very hard to go through.
So it's in Dutch, but on the PharmGKB website, you can find a link where you can also where you also can find the English version of the recommendations. That's one thing. If you want, I can also share that link with you, and then maybe you can distribute it. And the only thing, the limitation is that these are the warning texts and not all the background literature. That is in not so easily available. However, we for the first 10 years that this group was active, we were not allowed to share this in public, but there has been a policy change, and now we publish our results, our full results, as articles in the European Journal of Human Genetics. So there you can find all the background information and everything. And also there I would be happy to share some of those if that's helpful.
Yeah, thank you very much. We're coming close to the hour. Thank you very much, Professor Swen, for your time and effort. We learned a lot today about the PREPARE study, and we hope that in the future we'll learn more and more. Thank you very much.
Thank you all. Bye-bye.
Bye. Bye.